Alrighty, ladies and gentlemen, we have been talking about organic chemistry for the last couple of lectures. This one continues organic chemistry, finishes up the chapter for you. There is so much more to organic chemistry, but in high school chemistry, this will be your last exposure to it. We're going to be talking specifically about biomolecules now. So all of this chapter is organic chemistry, and organic means has single bonds with carbon. Not every organic molecule is involved in life. Some of them are formed by living things decomposing, etc. But here we're looking at specifically how does your body use organic chemistry. A lot of this will be review because in biology we talked about biochemistry as well. So this will be a lot like a lecture of the same title that you had in biology class. Okay, let me just give you a second to write that down. Okay, so we've talked about all of this before in biology. Biochemicals, biomolecules are huge. They're very, very, very big. And so they come in small units that we call monomers. A mere means a unit or a structure. And so a monomer is one unit. And then if you string a lot of these monomers together into a larger molecule, that's called a polymer, more than one unit. And so hopefully you're remembering this. Carbohydrates is the first category we're going to talk about. One sugar is called a monosaccharide, and that's exactly what the word means. Mono, one. Saccharide or saccharous is a sugar. So one sugar. So something like glucose or fructose is the monomer of that. Sometimes we call them simple sugars. And biologically speaking, nutritionally speaking, these are the things that your body can get energy from quickly. So if you eat a candy bar, or you drink some orange juice, or you have an apple, those are full of simple sugars. And the simple sugars go into your bloodstream, and your cells can immediately break them down and get energy out of them. So it's the kind of thing that you eat just before you're going to go do exercise. So those are simple sugars. Glucose, fructose are examples of those. You can string carbohydrates together, and then when you have multiple sugars, it's no longer a simple carb or a simple sugar. What's the nutritional term for lots of sugars trying to get? Complex carbohydrates, right? Sometimes you've heard of starches as well. So this is just an example of polymerization, right? You take two monomers and you join them together. And what happens, sometimes these are called dehydration reactions, because typically what happens is you take a alcohol group, a oxygen-hydrogen pair, off of one monomer, and you attach that open bonding site to a carbon, and you remove the hydrogen off of that carbon to get it to go. And so what winds up happening is one water molecule is created by the oxygen, hydrogen, the alcohol group leaving one monomer, and the hydrogen leaving another monomer, and then they get chained together, so we wind up with a polymer, right? One unit, one unit, one unit, now we have multiple units. Where's my mouse? There you go. Okay, so glucose and fructose, they're very, very similar molecules. They're both carbon and oxygen rings, and they both have alcohol groups hanging off the side. The only difference is the location of the oxygen bond. Is it between two methyl groups hanging off on a 5-member ring, or is it attached to a carbon that's in a chain and it's a 6-member ring? So if this oxygen were to pick up and bond with this carbon, then we would have fructose. Okay, that's the only difference. Glucose and fructose, same chemical formula. A polymer of a carbohydrate, there's two categories. There are some sugars that come in groups of 2, and so we call them disaccharides, right? Di means 2, saccharous is sugar, so disaccharides have 2 sugars in the molecule. And your body, not just your body, living things use disaccharides for short-term energy storage. If you have a little bit more sugar in your body than you need at this very moment, some of that sugar is going to be doubled up and made into 2 sugar molecules and stored, usually in the liver. Your liver tends to be the place where sucrose piles up a little bit, and your body waits to see if you need this in the short term. Now, if you've got a lot of sugar excess in a body, in your body, in a living thing's body, then it's not going to store it for a long time as disaccharides. It's going to eventually metabolize that and turn that energy into fat. And that's why if you eat too much sugar, there's more of you. And so, but the short-term energy storage, that, hey, I've got a lot of sugar right now, well, I need this in the next 20 minutes. Well, I need this in the next hour. Let's hang on to it and see. That's the disaccharides in your body. Sucrose is one example of that. There's lots of them, maltose, lactose, those are all disaccharides. And then living things can also chain together lots of sugar molecules. Hundreds or thousands of sugar molecules. And this is more often than not done by plants. Animals don't tend to build polysaccharides. That's mostly the domain of plants. And so plants will have an excess of sugar, and they don't get fat. You don't have fat roses, right? So when a plant stores energy long-term, it builds a starch. And the long-term energy storage molecules that are thousands or hundreds of sugar molecules long is called a starch. And when you eat certain vegetables, you get starches. What's a good source of starches? Potatoes, yeah. And that's why potatoes are a great food for long-term energy. If you're going to go on a run and you need energy right now, short-term, have some orange juice. If you're going to go on a marathon and you need energy long-term, have baked potato, right? Because there's tons of sugar in that potato, but it's all locked up in these huge starch molecules, and it's going to take your body some time to cut it back into sugar molecules so that you can eat it, so you can digest it. So it's like your body has a big long stream of sugar molecules, and it's saying, cut one off, there you go, have that. You need more? Okay, cut some off, there you go. Ready? Okay, cut some off. And you get all of these sugars slow released through your bloodstream as the potato is digested. So starch, long-term energy storage, and that's a plant thing. And then animals do use some long strains of sugar molecules structurally, but again, mostly plants that does this. Both of these are mostly plants. Long strings of sugar molecules called cellulose. And if you ever have celery, and there's that fibery stringiness to celery, that's cellulose. If you ever work with wood and you notice the grains and the fibers in wood, that's cellulose. The long stringiness of plants is cellulose. We can't digest that. Animals don't have the machinery to digest cellulose. So cellulose that we eat, because we eat plants, becomes what nutritionalists call dietary fiber. It's just stuff in your gut that helps everything move through, right? So that's where dietary fiber comes from. But both of these are sugar molecules that have been strung together, one in a way that we can digest and one in a way that we can't. Okay, so here's some examples of disaccharides. Sucrose is the most common. Lactose, this is what you get from milk. This is the sugar that's in milk. Okay, and then maltose is another common sugar that's in some grains and things of that nature. So this is, again, just 2 sugar molecules that have been stuck together. Okay? And then here's cellulose. So cellulose is a whole bunch of sugar molecules, thousands of them, in a big long string. And then these fibers of sugar molecules get woven together, and then those threads of woven together sugar molecules get then spun into like a sweater, right? And that like crocheted thing that's all sugar is called cellulose, and that's what plant cell walls are made out of, and plant fibers are made out of, and lots of a plant is made of. So the plant is all sugar, but some of it you can digest, some of it you can't. All right? Give you a second to write this down. Okay, the next section of biochemistry is lipids, and so carbohydrates are sugars, starches, and cellulose. For lipids, we talk about fats, oils, and waxes, and these are molecules that are used mostly in animals. Very few fat molecules in plants. There are some, usually as oils. Nuts have oils, avocados have oils, certain other vegetables and fruiting bodies have oils, but most of the time, fats, oils, and waxes are an animal thing as opposed to a plant thing. The monomers we call fatty acids. It sounds like something a middle schooler might have called somebody they didn't like. You're fatty acid. But fatty acids are still rather large molecules. So when we say monomers of sugars, sugars are pretty small. And you string thousands of them together to make starches and cellulose. Fatty acids are already pretty big. A fatty acid molecule is already 25, 35, 50 carbons long. So it's already a pretty big molecule. And it ends in a carboxylic acid group at the end, which is why it's called fatty acid. And these things are used for long-term energy storage in an animal. So a plant has too much sugar, it makes starch. An animal has too much sugar, it makes fat. And the fat is more dense energy storage. You, pound for pound, you get more calories out of fat than you would out of a potato, right? If you had a 1 pound potato, that's a lot of calories, but it's something that you could reasonably burn off in a day. If you have 1 pound of animal lard and you were to eat it, that would be gross. But if you were to, you would not burn that off in a day. That would turn into fat for you. And so a lot more energy, a lot more densely stored in a fat molecule. Fatty acids are non-polar. You will not get a fat to mix with water. Oil and water is like the famous it doesn't mix example, right? Fats and water don't mix. And so they tend to be used well for membranes and cells because the water will stay over there or stay over there and not cross the membrane very much. So that's one way that your body uses it. A polymer of fats, most of the time, is called a triglyceride. Triglyceride, glyceride is another word for a fat molecule. It's a particular kind of fatty acid. Glycerin is one kind of fatty acid. So 3 glycerin molecules joined together make up the most common kind of fat polymer. So I'll show you a picture in a minute, but 3 long strings and their heads are tied together. The heads are tied together by a molecule called glycerol. And so you have 3 fatty acid chains joined together by glycerol. We call it the triglyceride. Okay? There are 2 kinds of fatty acid polymers. And nutritionally, they're significant, and we've already kind of talked about this when we talked about alkanes, alkenes, and alkynes. A saturated one has all single bonds, and saturated fats, we call them saturated because you can't add more hydrogen to it. It's got all the hydrogen it can hold. It's saturated with hydrogen. And these tend to be solids at room temperature, fats and waxes. Inside a mammal, fats are kind of squishy. As you can determine, if you were to squish it yourself somewhere, the fats are kind of squishy because you're warm, and so the fat is pliable and soft. And if you got it much hotter than room temperature, than your body temperature, if you're talking about 165 degrees that human fat melts, then it would liquefy. So, and you see this hopefully not in cooking human meat, but if you cook other kinds of meat, right? When you put it on the grill, it's solid, and then it gets hot and it gets runny, and you get gravy that comes out of cooking meat, right? The drippings, and that's the fat that's liquefying at above 165 degrees, and that's why those juices form in your pan, right? So saturated fats are solid at room temperature. Unsaturated fats tend to be liquid at room temperature. So these are your oils. This is why it's a lot better to fry your eggs in olive oil or avocado oil or grapeseed oil than it is to fry your eggs in butter. Butter is a solid at room temperature because it's got a lot of saturated fat. Oils are liquids at room temperature because they're not saturated, they're unsaturated fats. So your body can digest this nutritionally a lot better than the saturated fats. But the only difference is the number of, or the existence of double and triple bonds. Your body can break down double and triple bonds a lot easier. So a saturated fatty acid is all carbon and hydrogens. And this is only drawn, I didn't count it, but I'm going to guess like 20-ish carbons. Most of the time, they're much longer than that. An unsaturated fatty acid has a kink in the chain. It bends at the double bond. And the double bond is where your body would start to break down the fat. So if your body's looking to turn some fat into energy because you're exercising, you've spent your sucrose, you've spent your glucose. It says, I need more sugar, I need more energy. I'm going to go take apart some fat. It'll start eating this fat molecule at the double bond. Double bond can be anywhere in the chain, but it'll start there. And then once it's started, it can take it apart as it goes. But something like this, it doesn't know where to start. It looks at that and it's like, yeah, I give up. Stack that to the side. Find a molecule with double bond. And that's why you will digest the unsaturated fats in your body much faster than you will digest the saturated fats, because these are left until last. They're harder to break down. Okay? This is a triglyceride, 3 fatty acid molecules strung together by a glycerol molecule. And these are unsaturated fatty acids because they've got kinks in the chain. They're not straight. So this would be an unsaturated fatty acid. A saturated fatty acid would have 3 perfect strings coming off of the glycerol molecule. Okay? And the more unsaturated they are, the more twisty they get. You can have lots of double bonds or triple bonds where they get all proteomorphous. But that would be easier still for your body to break down. Okay? Steroids are another example of a fat. Steroids are fatty acids that have been made into a ring. So you take this string of carbon and hydrogen and you loop it back on itself and glue its head to its tail, and you get a ring. And the different numbers of carbons in the ring and the different numbers of rings makes the steroids different from each other. But it's a ringed fatty acid. And these are used by your body to control various aspects of your body. They're messengers, they're chemical messengers. If your body needs to do something, it'll release a hormone. And then when that hormone gets to your cells say, oh, I'm supposed to do this. And those hormones are steroids. So the hormones are fat molecules. But the way that they're built is interpreted by the cells of your body as a signal. So testosterone is a hormone. Testosterone starts to be produced in a human embryo a couple of weeks into gestation. And initially, every embryo starts off developing into a girl. It's kind of interesting. Every embryo starts off developing towards being a female. And then I think it's at 12 weeks, 8 weeks, somewhere around there, the embryo starts to produce testosterone if it has Y chromosomes. And then the embryo's like, oh, just kidding, I'm a boy, right? And the structures that were going to be ovaries all of a sudden change what kind of cells they're going to make and descend and become testes, right? And the tissues that were going to become the uterus and all of that stuff, instead of being sticky innies, become sticky outies, and other parts form. And so the embryo, though, every embryo begins building female parts until the testosterone starts to be made. And then it's like, oh, my bad. And it changes what it's going to be. There are only incredibly rare circumstances, like less than 1 in 100 million, where an embryo is genetically male, has Y chromosomes, but the testosterone is not made in enough volume, a large enough volume, where the embryo changes its development. So, again, incredibly rare, like maybe 1 in 100 million, something like that, where a person will be female with Y chromosomes, and that person will be sterile because eggs that are produced with a Y chromosome won't be fertilized, won't make healthy offspring. So that person, then if they were to go get genetically screened, they are a girl, right? They go get genetically screened and for some other reason, or maybe to figure out why can't I have kids. And the doctor would be like, oh, so actually, your genes say you're a boy. But it's incredibly rare, and this is not a reason to be like the whole transgender bathroom thing. No, that's not what we're talking about. Those people would have no idea that they have Y chromosomes until some geneticist told them that. Their body they would be normal people, normal ladies. But almost, every time, it's not worth looking at 1 in 100 million. The testosterone is produced, and the chemical creates a change in the body. Other steroids, like cholesterol, are not used as chemical messengers. They actually kind of get in the way. Some cholesterols are useful for heart health. The high-density lipids are useful for heart health. They do good things for you, and then the LDLs, the low-density lipids, they float around in your blood and clog things up. So this is also a steroid, but it's not used as a chemical messenger. There's tons of them. Caffeine is another one. Vitamin D is another one. There's tons of chemical messengers in your body that are steroids that are just fat molecules joined into a ring. The last group is proteins, and again, I'll pause here. Okay, so the last group of them, last group of biomolecules are proteins. Proteins, the monomer, the small molecule, is called an amino acid, and amino because it has a nitrogen group, and an acid because it has that double bonded oxygen OH group. So amino acids are named what they are because of the components, the functional groups in that molecule. These are pretty small. Amino acids are pretty small. When you see a molecule of amino acid drawn out in the scheme of biological molecules, they're not big. But you string hundreds, thousands, tens of thousands of them together, and you get very, very large molecules called proteins. So a protein is a string of amino acids. We sometimes call them polypeptides as well. The reason being that the sort of bond that exists between two amino acids is called a peptide bond. So when a molecule of leucine and a molecule of, I can't think of another one. Yeah, I'm going to say two lysines because I can't think of any another name of an amino acid right now. Two lysines get together, they kick out a water molecule between them, and they join, and the particular way that they join, that's called a peptide bond. Don't worry about it, but that's the name of that bond. And so when you string a whole bunch of amino acids together, you have all of those peptide bonds. Sometimes proteins are called polypeptides, more than one peptide bond. And they can be thousands of amino acids long. Huge, huge, monstrous things. And when we went through biology last year, we studied how they're made with the polymerase molecules and the ribosome and all of that kind of stuff, transcription, translation, and all of that process we went through in biology. So this is, again, just a reminder that amino acids join together to make proteins. And then the order of the amino acid is just one level of information. The amino acid is unique, the protein is unique not just because of its order of amino acids. It's not a thread that makes up a protein. That thread of amino acids is now folded a whole bunch of times in very complex ways to make a three-dimensional shape of this object that is a protein. And then usually multiple proteins get together to fit within each other and stack next to each other in a particular way to make a protein complex. And it's usually protein complexes that your body uses for a task. And so it's more than one protein, and each protein has to be folded the right way to fit together so that they can all do their job and make some kind of machinery happen. It is one of, I think, the best examples of intelligent design out there, because the levels of information is incredible. There's one level of information for what's the order of the amino acid. There's another level of information of how do we fold that amino acid chain. And there's another level of information of how do these get together and work cooperatively. And each protein will be made by a different gene. And so in order to get the right protein complex to actually do the job the cell needs it to do, there's multiple genes involved to make these things fold properly so they can fit together, and then to fit them together. And it's random chance. It's just stupid to believe in random chance. That's going to produce this. So structural, they're used for structural molecules, and they are enzymes that build, enzymes that carry on, biological reactions. So carbohydrates are mostly a plant thing. Fats, mostly an animal thing. Both plants and animals use proteins, although animals, more of the body of the animal is made out of protein, and more of the body of the plant is made out of carbohydrates. But still, this will be found in both, advanced domains of life. So again, looking at the nature of a protein versus an amino acid, an amino group has a carboxylic acid group and an amino group, and then they call this the R group. It's just the rest of the molecule. There'll be another couple of carbons and their stuff stuck on it. But the amino is right next to the carboxylic acid, which is why we call it an amino acid. And these come in several different flavors. Amino acids, there are, I think, about 20 or so, 23 amino acids, different kinds of amino acid. And the order of them controls what the protein is able to do, and then how the protein is folded controls what the protein is able to do, and how those folded proteins then also interact with each other. So this is an example of what I was talking about before. It's my picture's in the way. There you go. String of amino acids matters. The way that amino acid itself is wrapped or folded, its own self matters. And then how these interact with each other, multiple proteins functioning together to make a complex of proteins. That's the thing that your body needs to do some work. It's incredible the amount of information in there. Okay? And I think that is it. Oh no, there's one more. Just kidding. Last one. Now I promise it's the last one. Nucleic acids is the last kind of biomolecule. So, and this is in every living thing. Every living thing contains DNA or RNA. The monomers of these things are called nucleotides. They are nucleic acids. They're also carboxylic acid groups, but differently arranged, so they're not an amino acid. But similarly to proteins carrying information in ordered amino acids, that information comes from ordered nucleotides in DNA or RNA. So one nucleotide, there's only 4 kinds: adenosine, guanine, cytosine, and thymine. And these 4 small nucleotide molecules arranged in order form RNA or DNA, depending on the organism. And that ordered sequence of nucleotides carries the information for how to make and operate a living thing. So the instructions for how to build La Villa is contained in the DNA of every cell of Lavilla's body, and it's very, very, very similar, but slightly different from the information to make an alec. And so if you were to compare their DNA, almost all of it's the same, but there are slight differences, which is why Lavilla and alec look remarkably like each other. 2 arms, 2 legs, 10 fingers, 10 toes, 2 eyes, nose, etc. But slightly different things, right? And how to carry on the chemistry that makes that living thing function is also encoded in the DNA. So ridiculous volumes of information. The Human Genome Project took years and years and years and years to sequence one person's DNA and to write down all the letters in order of what that person's DNA was. And the volume of data that was generated was stupid. If you were to print it and put it in books, you would have to have a building something like the size of the Smithsonian Institute to hold the printed information of one person's DNA. It's crazy how much information is there. And so DNA is, again, another remarkable testimony to an intelligence behind the universe. The guy who headed up the Human Genome Project, his name is Francis Collins. And Francis Collins began his work as an atheist, and the process of sequencing the human genome saved him. He wound up being a Christian by the time he was done, because when he was done with it, he said, there's no way that this is a mistake. And so he is now a very outspoken proponent of intelligent design and of God behind nature, right? It's really cool. So the DNA sequence is just an astounding amount of information. A short segment of DNA that codes for a particular trait, we call a gene. We covered this in biology. This is all review. Several genes, thousands of genes worth of DNA or RNA will be put into one molecule that's wound up into this little thing called a chromosome. Tens of thousands of monomers of, or even millions of monomers of nucleic acids in there, okay? Most organisms have several chromosomes. And my last one fell off the bottom. Humans have how many? How many chromosomes do we have? We have 23, yes. 23, and each of them have a friend. They're all paired, so 23 pairs of chromosomes. And so this is, again, an astounding amount of information and a great reason to praise the Lord. Here's one example of a nucleotide, okay? There is still a lot of nitrogen in it. As a matter of fact, there's two 6-member rings, or sorry, a 6 and a 5-member ring that are mostly nitrogen, that have a lot of nitrogen in it. But it's not immediately adjacent to a carboxylic acid group, and so we don't call it an amino acid. It's a sugar, and the sugar that is used here is ribose, and then usually it's ribose missing an oxygen where there would have been down here, and so they call it deoxyribose. It's missing an oxygen for normal ribose, which is why a nucleic acid hooked up with deoxygenated ribose, deoxyribonucleic acid, that's where DNA gets its name, right? And then RNA has that missing oxygen, so it's not deoxyribonucleic acid, it's just ribonucleic acid. And so this is a nucleotide. You string whole stupid amounts of them together, and you get DNA, right? And that is an amazing testimony to the mind of God. There you go, biomolecules, almost all review from biology. Do you have any questions? Anybody?